Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled Respiratory Drug Development, Comprehensive Strategies to Improve Data Quality. My name is Ryan Muse and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit your questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who have registered on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading provider of endpoint data solutions to the clinical trials industry, generating high-quality clinical evidence for life sciences companies. They offer comprehensive evidence generation solutions that combine medical imaging, eCOA, precision motion, cardiac, and respiratory endpoints. Now I would like to introduce our speakers for today's event. Dr. Todd Listine brings over 25 years of experience spanning pulmonary and critical care clinical leadership and pharmaceutical medical safety. At Clario, he leads respiratory medical science with a science-first approach to supporting customers in advancing respiratory research. Kevin McCarthy has over 50 years of experience in lung function testing, including 4 decades leading pulmonary function laboratories. He is a recognized expert in pulmonary function quality with extensive publications and contributions to ATS-ERS standards for spirometry and lung volume testing. Dr. Martha Matthews Martin is an education and workforce development leader specializing in innovative training strategies for pharmaceutical and clinical research organizations. At Clario, she leads a global team delivering scalable, customized training platforms tailored to client needs. And Dr. Phil Lake has over 25 years of experience in respiratory drug development, supporting global clinical trials across a wide range of therapies and respiratory diseases. He is committed to advancing understanding of variability in lung function testing and how study design and ongoing active management of data quality via plausibility review can help identify hidden quality issues which undermine study outcomes. But now, without further ado, I'd like to go ahead and hand the presentation over to our first speaker for today, Dr. Todd Listine. You may begin when you're ready.
Thank you, Ryan. Go ahead and advance the slide, please. And you can advance again, please. Next slide, please, Ryan. There we go. So greetings, everyone. We really appreciate the opportunity to present to you today. Thankful that you could join us. Here is today's agenda. I will be opening with a sort of a general introduction and discussion of centralization of data collection and risk-based monitoring. I will then turn it over to Phil, where he'll speak a bit about study and site level practices that can optimize data quality in respiratory trials. We will then hone in a bit on actually test level practices at the bench side as to how we can contribute to optimize that outputs there. And Kevin will provide that information, and then we will shift to Martha, who will then talk specifically about site and technician training in towards the goal of optimizing the quality of spirometry. Next slide, please. Next slide. So when you responded to our invitation, you're offered a survey question regarding what you, the audience, viewed as most challenging in generating and maintaining highest quality respiratory data. You can see that by your responses that the spirometry maneuver itself loomed large, identified as most impactful of those we offered there. This was followed by management of variability of the data. It's interesting to note that bringing up the rear, it was the concept of effective site training. We're hoping we can elevate that value in your minds, as getting this right as you initiate your trial can go a long way towards avoiding both variability and poor quality of the spirometry itself. Next slide. So my topic is that of centralized data collection and risk-based monitoring in the pursuit of optimized data quality. Go ahead and advance the slide again, please, Ryan. Though aligned with the survey results, let me start with a statement that performance of the spirometry maneuver itself can be quite difficult. Requires optimal coaching, cooperation on maximal effort from the tested participant, requires active assessment of the emerging quality of the test during the actual conduct of the trial with each effort. And this is inclusive of appropriate in-stream prompts to improve performance. There's a necessity for assessment of repeatability and other parameters to meet ATS ERS standards. Applying these standards can help improve quality, but certainly does not ensure it. In fact, our experience is fraught with examples of high ATS ERS graded data that remains of poor quality, data that's not reliably representative of an individual's true pulmonary physiology. It's very important to note that the standards themselves were written with the healthcare environment in mind. Data may be supportive, but perhaps less critical in that space, given the availability of other data that help inform treatment decisions. These include things like symptoms, observations offered by loved ones that may be accompanying the patient during a visit, physical exam findings, and others. For clinical trials, the emerging data point that comes from a spirometric measure may actually be the primary endpoint. So it's quite critical that that be accurate and appropriately representative of the patient's true physiology. This is further compounded by the fact that the clinical trial may include tens or hundreds of sites and hundreds or more of study participants. There's lots of risk for problematic data quality coming from this, and indeed a great deal of variability. So what can be done during conduct of the trial to assure recognition of emerging problematic data in the first place, and hopefully recognition early enough to offer opportunity for intervention prior to meaningful impact on the resulting data set? Next slide, please. Next slide. So this is where centralized data collection and the application of risk-based monitoring comes in. So I want to briefly cover this, but very quickly transition to the actual concepts and applications we have found that can be practically applied to assure recognition of emerging issues and indeed offer opportunities to intervene. FDA has issued a guidance document, which was then followed by a Q&A document on this topic. This first was released on 2013, and the Q&A in 2023 as well to follow up. Both of these documents articulate acknowledgement of the increasing number and complexity of trials, which increases the demand for more efficient and streamlined means of monitoring. They also call out use of electronic systems, which may add a bit of complexity, but also creates an opportunity to more effectively and efficiently do so, given current technologies. Although there is no specific guidance on how to pursue these activities, done properly, there is recognized opportunity to focus more intensive oversight towards data at highest risk, resulting in sufficient vigilance without excessive effort. The goal is not only appropriate oversight of subject safety, but also that of data integrity. Next slide, please. Here, I've pulled a few excerpts from the documents themselves to provide some insight on the FDA's perspectives on this. You can see that they emphasize prospectively identifying that data that is critical, not only to assurance of participant safety, but also data integrity, as I've mentioned. They acknowledge the opportunity to recognize anomalies in the data, even calling out fraudulent and fabricated data. They call out non-random data distribution to be watched for. They emphasize routine review of data for inconsistency and outliers. They propose the value of applying statistical tools to help flag data that is worthy of more focused attention and analysis. These can include problematic data distribution and data showing implausible degree of variance. Next slide, please. And finally, I share top-level findings of a recent report, the Tufts Center for the Study of Drug Development Impact Report, that speaks to the general acceptance and uptake of the application of centralized data collection and risk-based monitoring. It is encouraging to see that at least half of the small sponsors and 3/4 of large sponsors are embracing implementation. More than half found that data integrity and quality to have at least be somewhat improved as a result. And reassuringly, this is not at the expense of, but instead reports significantly improved efficiency of their monitoring visits. And in fact, site relationships have improved in terms of quality. So I'll close with that. We'll now move on to the study and site level applications that may be in play here. I'll turn this over then to Phil Lake to cover this issue. Thanks so much.
Thanks very much, Todd. So I'm going to, as Todd said, I'm going to move on to talk some of the site and study level approaches that we take to try and identify data quality issues. So I thought I'd start off with what is, an idealized trial, if you like. So this is a COPD trial, and it has serial spirometry across a number of visits. So what you can see here is that at each time point you have really nice reproducible data. And again here, for each individual visit, you can see the serial measurements along the bottom, gradually improving over time with a little plateau. And then each subsequent visit has a slight improvement of the trough values at baseline as you start the visit. So really, really nice, plausible, predictable patient transition. So again, an ideal set of data. Unfortunately, reality isn't quite like that. So what I've generated here is an example from an asthma study we ran a number of years ago. And again, now this shows you longitudinal variability of FEV1. And each of these lines represents an individual patient. And then you can see the sequential visits along the bottom here. So we start off with a visit 1 pre and post measurement. And then we go through visit 2 to 4, which is the run-in, which are all pre-measurements. And then you get randomization and move through to visit 10, which is the endpoint. So really change from visit 4 to visit 10 is the study effect we're looking for. Now I've highlighted one of these patients here in blue, and you can see here this kind of real roller coaster effect of their lung function over time, which really isn't normal. And when we look at this individual patient, we can see a screening value, a pre-value of just above 2 liters, and then we have a post-bronchodilator value above 3.5, which is a massive treatment response. So that in itself starts to raise eyebrows. So you start to think, well, is this really the trough value? This one presumably is a post-bronchodilator response, as we'd expect. But when we come to visit 2, we can see a very similar value to the screening post-bronchodilator value. This is supposed to be a pre-bronchodilator trough measurement. So again, it raises in your mind, well, have they actually taken their bronchodilator? And it's just not been recorded. We then see a drop back to visit 3. Again, very different from the visit 1 trough. And then we have a visit 4 trough. So when you look at these 3 measurements, you're thinking, well, which one is the most accurate, really? It's unlikely that this is actually variability in disease, because there is a requirement for stability in run-in, and there's no real change in treatment to actually drive this. So this is one of the real problems we had. So when we start to look at this data, we notice that some sites have much more variable data than others. And so what we decided to do was try and quantify the variability of a site's data. So to do that, we looked at the standard deviation of on-therapy FEV1 change. And then because we've got some patients with 3.5 liter lung function and some with a 1 liter lung function, to be able to compare that, we actually then express the variability as a percentage of their baseline value, and hence we get the modified coefficient of variability. So let me move on to show you a little bit more. So when we express the modified coefficient of variability, I just want you to look at this chart here now for the time being. So we can actually see each patient represents an individual point here. This is the percentage modified coefficient of variability, so from 10 up to 60%. And we can see each vertical line represents a different site. So when we look at this one site, this is some of the worst data we've come across in a clinical trial. So you can see that some of the data is really highly variable. If you compare this site to some of these others, you can see that they have, almost consistently lower variability data, maybe with the odd outlier in this case. But again, you can see a big difference. However, when we look at the ATS-ERS compliance of all of this data, we can see that only about 1% of the data is classified as unacceptable. Now, if you compare that with the rest of the study, we can actually see that the quality of this site, when you're just looking at ATS compliance, appears to be better than the rest of the study. And yet the variability doesn't point to that. And when we start to look at these highly variable patients, we start to see real problems with the technique and other issues. So moving on to I wanted to basically say that ATS 2019 is actually a great approach. I'm not saying that ATS isn't suitable for actually working out lung function quality. It is. And so the broad premise is that if you have a consistent technique for doing a force spirometry and the data is reproducible, it's more likely that the data is accurate. And that still stands true. However, technique doesn't guarantee that you have accurate data. So I want you to consider these scenarios. So if you imagine you bring in a patient to test them and you select the wrong patient from your list, so nobody knows their patients by 103, 453 or 454, you know them by their name. So it's fairly easy to select the wrong number. Now you can actually do the testing, all the ATS compliance will be perfect, and therefore the quality will be regarded as acceptable. Unfortunately, it's just the wrong patient. And so, the actual accuracy of the data is completely undermined, but the ATS compliance is still good. Again, another example, if you look at the medication washout, now you remember the ones I just showed you in the last study, where we see highly dubious data with a pre and a post being virtually identical. That happens at various degrees. So again, the ATS compliance or adherence won't actually pick up the medication washout variability. And so this is another area which isn't covered by this. It is also possible, and Kevin's going to show you this in a little while, to have consistent submaximal inhalation. So repeatability doesn't mean it's accurate, it just means it's consistent. And so it is possible to have highly reproducible data, and there are instances within the ATS criteria where you're basically, you can grade data as acceptable, even though it's not technically accurate. And again, this brings us into this other category of site modification of the technique. So there is the FIVC, FVC comparison to try and work out whether you've reached total lung capacity before you exhale. And we are seeing more and more sites actually modifying their technique to make those values align rather than using it to check for full inhalation. So Kevin's going to go through that in some detail. And then finally, there's a couple of other things we use in terms of implausible changes to lung function. So we do look longitudinally at data to help try and work out whether transitions make sense. We also look at things like post-bronchodilator effects within a patient. So if you take the reversibility, so if you have an increased FEV1 response to a bronchodilator, you should also see a corresponding response in the FEV1 FVC ratio. So as you open up the small airways and improve FEV1, you should also see an improvement in obstruction. And when you actually start to plot those together, you can see big improvements in FEV1, but actually a worsening obstruction. Now, this generally relates to either a switch in patient or some other core technical issue. And then finally, we use biological plausibility or implausibility. So if you're testing a 70-year-old asthmatic, for example, and they manage to exhale to a plateau in 3 seconds, and you've got a nice rounded profile, likely is that that's not the patient you think you're testing. So you've probably made a switch in the patient inadvertently. So finally, a little bit of data to express the extent of this implausible data within clinical trials. So we do see that up to 23%, that's the highest we've seen in any trial, of the data can be classified as implausible. So let me just show you what I mean by implausible. So another way to present the data, so in this chart, I've actually shown the change from baseline to the current visit. So this is either improvement or decline in FEV1 plotted that against the variability of post-randomization FEV1. And so again, you can see in 20, 30, 40% variability. Once you get above about 15% variability, we start to see a preponderance of these really high change from baseline data points. So they don't have to be high improvements. They can be declines as well. But effectively, it really depends on the high variability. The fact that you get an improvement or decline really depends on what patient is impacted by that variability most. So again, what time point. So you can have very big negative values or big positive values. In general, we tend to see increases because of the nature of the calculation. But again, if you take up this section of data here and look at the average FEV1 change and compare it to this area where it's considered to be more normal, we can get 10 to 30 times higher levels of mean FEV1 change than this population. So within a study basis, I mentioned 23% before. It normally varies from about 6 to 23, but the critical range for most studies is something between 8 and 10%. So there's still about 10% of your data, which potentially is carrying forwards inaccurate data, which is impacting your variability and your treatment responses. So if I've not already persuaded you about this, I have one last slide to try and do it. So this is a case study where we've looked at, again, at another ASPA trial. We've taken the most variable data in the trial and plotted this all out. So you can see 23 to 40% variability. Over here, you can see the initial ATS-ERS compliance of that data. So it was all either borderline acceptable or acceptable. So, what we consider to be good data. If you look at the magnitude of change, so everyone changed from baseline, you can see these are really, significant changes. This is like having a lung transplant. So this isn't a normal treatment response. And again, so if we actually then start to look holistically at the data and say, well, what evidence is there for submax inhalation errors or patient switches, we then see that about 85% of this data is rejected. So we have two patients even where the patient who started the test wasn't actually the one that finished the trial effectively. So we switched the patient halfway through. There are two, you notice, which have acceptable data still after this review. But just remember back to the point is that, again, the technique doesn't pick up variable bronchodilation. So these tend to be smaller values, but these could still be impacted by a patient not actually adhering to the washout properly. So I'm going to hand you back over to Kevin now, who's actually going to take you through the mechanisms behind how this kind of thing happens. So thank you very much.
Thanks, Phil. Great review. Next slide, please. So yeah, I'm going to be talking about testing. I think that's where we're going to end up landing when you're talking about, Phil's done a nice job of telling you how we can detect problems. What I'd like to do is talk about preventing the problems. So what you see in front of you here is a volume time tracing on the left showing spirometry and how it's changed since the 2019 spirometry standards were written. We start off with just 3 normal tidal breaths, resting breathing in this particular case. And then the operator then coaches the patient to rapidly inspire to full inflation and take them up to the second phase. That's the start of the forced exhalation. Let's pause there for just a moment because that's a point that's a little bit nebulous. That is, how does the operator know exactly when to tell the patient it's time to start that forced exhalation? Because they're vigorously coaching. Deep breath in, more, blow. What triggers the command to blow? If you look at the guidance given in the ATS standards, and there's been 5 set of standards since 1978, all of them said forced exhalation should start from full inflation, but none of them until 2019 gave any guidance on how to do that. And the guidance in the 2019 standards is look at the patient and look for, obtain feedback from them that they're really at full inflation before you give that command. And I'll touch base on that just in a little bit. So at phase 2, the command to blow is given, and hopefully the patient does a hard, forceful exhalation, and the operator has to continuously coach more, because patients feel like they're empty before they really are. And what the operator is doing is taking them to phase 3, the end of the forced exhalation. At that point, the operator either seen that the volume isn't changing and they can see the patient and show that there really aren't, they are pushing, but no more air is coming out. Or, as is the case in most obstructive lung diseases, the patient is continuing to push and air is still coming out, albeit at slower flow rates. And in that instance, the recommendation is until you see the plateau, try to take them to 15 seconds. Once you reach that point in time, again, another transition, and operators have to have the ability to keep talking through the whole thing because they transition from more, a little bit more. Now, big breath in, deep and fast, a little more. And they take the patient again up to full inflation. So this fourth phase is brand new to the standards. It was added in the 2019 standards. Prior to that, it wasn't considered to be a requirement. And a lot of people are a little bit confused about what's an inspiratory parameter have to do with spirometry when in fact we're looking at expiratory parameters, in particular in clinical trials, the FEV1 and the FVC. So visually you can see what it's there for. It's meant to compare the volume that they inhale from the end of forced exhalation up to full inflation to what they just exhaled. And since they haven't come off the mouthpiece, the end of the forced exhalation is also the start of the forced inhalation. And you can see here in the example on the left-hand side, both of those volumes are equal. And that's evidence that supports the notion the patient really was at full inflation. Now contrast this with the image on the right, which shows a discrepancy. There's a gap there. They breathed in more at the end of the forced exhalation than they blew out. That gap there represents what we call submaximal inhalation error. And in this case, the patient was given the command to start too soon, and that's what we want to address. Next slide, please. So it's actually quite common, and I'll share with you some information on that in a second. And I have some theories about how it came to be. So if you give the command to blast the air out while the patient is still inhaling, as we often see on these volume time tracings, you virtually eliminate back extrapolated volume errors. That's back extrapolated volume is air that's exhaled with less than maximal force. And it usually happens when you take a patient up to full inflation and hold them there too long. It's difficult. The lungs and the chest wall both want to recoil back and they let a little bit of air out. So it's the most common start of forced exhalation error. And so you virtually eliminate that by giving the command to blow before they stop or reach full inflation. There's also what I'll call the repeatability myth, and it goes like this. You can't get repeatable FVCs and FEV1s unless both efforts were started from full inflation. It's absolutely a myth, and I'll show you some data to support that. And the FIVC, keep in mind, it's new. So if people were just evaluating their own technique, for example, or they're participating in clinical trials and getting feedback from overread, they would see, oh yeah, I'm doing a great job. I'm getting acceptable FVCs and FEV1s. The data is repeatable. End of story. I'm good. But you add the FIVC to the mix. You add the evaluation of full inflation, and sometimes you're revealing that that data is not something you want in the clinical trial, that it didn't start from full inflation. And then add to all of this is that spirometry is by and large taught by word of mouth. Joe teaches Bob, teaches Mary. And if one of them, if Joe has bad technique with respect to coaching to full inflation, then it's likely to be passed on to the others as well. Next slide, please. So this next slide shows a lot of things that we've presented over the years as posters at the various scientific meetings, ATS and ERS. And it's a lot of what we've learned about submaximal inhalation error. So to support the notion that it's common, one of the things that we did, and Phil alluded to this already, he said from 6 to 23% of the data is implausible based on the tools that we have. And this is one of the ones. This study showed 13% of the data, not 13% of the data, 13% of 7,000 measurements that really, when viewed through the lens of the 2005 standards, looked perfect. But when you added FIVC to the mix, you found out that the forced exhalations didn't start from full inflation. And those measurements came from the majority of the operators at the majority of the sites. Not to say those operators, every measurement they submitted had submaximal inhalation error, but it's coming from 69% of the operators at 81% of the sites, so it is prevalent. And when you couple that with some starting from full inflation, that's the kind of variability that we see in clinical trials. We also looked at the magnitude of the submaximal inhalation error, and we found that on average, the efforts flagged for SIE were starting 0.27 liters plus or minus 0.15 below full inflation. That doesn't sound too bad unless you look at it in the light of what's considered to be a fairly decent treatment effect when you're trying in a clinical trial for asthma. It's about that, and so it certainly is a big chunk of that. And in each one of those 3 trials that we pulled the data from, there were outliers where the SIE exceeded 1 liter, and those outliers can really sort of exceed the contribution to the overall picture of the data. They really add a lot to the noise. And we've said that SIE is the major contributor to variability. We had one opportunity early on to really test that, and we looked at 2 studies with the same disease indication and severity. They were the same protocol, and one was using the 2005 standards, the other study was the 2019 standards. And at that point, it was one of the earliest studies where we were using the 2019, and we had strict adherence to the FIVC rule. And by that, I mean those measurements were being called unacceptable. And when we compared those 2 studies, the 2 measurements that were required for the baseline, they were measurements done 30 minutes apart. There was a 51% reduction in the variability between those 2 measurements in the 2019 study. There was a 45% reduction in the modified coefficient of variation that Phil was talking about. That's the variability of the measurements on therapy. And then for those that are flagged, patients that are flagged for high modified coefficient of variability of the FEV1 while on therapy, there was almost a 90% reduction in the patients that showed that. And when you look at those 3 bullet points, you kind of come away with a great appreciation for the problem that submaximal inhalation error represents. And you might think that we've solved the problem, but not yet. Let's go to the 4th bullet point. So the 4th bullet point is a work that we presented in 2024 at the ATS that showed the comparison of the FIVC and the FVC is not always reliable. Next slide, please. So these 3 examples here come from clinical trials, and they're the one on the left, the panel on the left shows pre and post bronchodilator. This is typically done during screening to show that the patient, an asthmatic patient typically, the protocol wants to show that they have at least a significant response, a 12% increase in the FEV1 or 200 ml, whichever is larger, or now a 10% increase in FEV1 percent predicted. That and this patient certainly did have that. If you look at the curve on the left, it really is not showing all that much obstruction. There's really only a modest degree of concavity in the expiratory flow volume curve. Yet if you look at the magnitude of the response, both in volume and the flow the airways are supporting during the exhalation, this is many standard deviations above the expected response in an asthmatic. And the reason, you don't have to look too far to find out how that could be. If you look just below the flow volume curve on the left, you'll see a volume time tracing showing the forced exhalation started from a tidal breath. And then it leaves the question about well, in this particular effort, the FIVC that followed the FVC matched in volume, meaning the expiratory and inspiratory flow volume curves show the same excursion along that volume axis. But notice this, notice that the arrow pointing to the inspiratory flow volume curve is really pointing out that the flow rate during inspiration is only about a half a liter per second. And decades of experience tells me that no patient will want, completing a forced exhalation is going to want to inhale that slowly. So clearly there's some instruction or guidance being given while that's occurring, and it's likely that this was just stopped by the operator, show good matching of the FIVC with the FVC. So clearly not the intended use of that comparison. Let's move to the middle panel, and we see this is showing a really robustly inhaled FIVC. Look at the The shape is what we call the upside down haystack appearance. And that's typical of an inspiratory vital capacity being inhaled with maximum effort. However, there's a problem. Even though that FIVC matches in volume, notice that where it stopped, flow was still at 2 liters per second. This occurs when an operator hits the end measurement button. So looking at it on a data table, you'd say, well, that FIVC matches the FVC, must have started from full inflation, but in actual fact, here's evidence that it didn't. All of this, these are 2 different techniques for adjusting the collection of the FIVC to make the data quality look good, when in fact, and what this results in is what you see in the rightmost panel. Those are 8 efforts from the same measurement. All 8 efforts had the same end of forced exhalation. From the start of the forced exhalation to the very end, where they started the inspiration of the FIVC, was 15 seconds in every effort. And if you look at the FIVCs here, so they're all starting the FIVC from that same state of emptiness, but the volume of the FVC, the smallest one compared to the largest one, is about 1 liter. And each one of those efforts shows a matching FIVC, with there's some evidence that the mouthpiece is actually being pulled out of the mouth. You can see on some efforts there's a slight increase in inspiratory flow before a very abrupt end of inspiratory flow. So all 3 examples of something that is really confounding our efforts to sort of characterize improperly performed data, submaximal inhalation error. Not that all submaximal inhalation error data is bad. Let's go to the next slide and I'll expand on that notion. Can you pass this one too and go to the next one? Okay, thank you. So this is a pie chart, and it's these are measurements taken from a clinical trial for asthma. And let me explain what those different segments of the pie represent. So we see 57% of the measurements falling into the green category. Green means that the FIVC volume suggested the forced exhalation started from full inflation, and the FIVC was inhaled vigorously. That is, we have a way of characterizing whether it was inhaled with maximum effort or not, or approximating that, or getting a good idea if that occurred. So 57% of the data, if I was a sponsor looking at that, I'd say I have high confidence that that data really represents the best that patient could do on that day. So the purple wedge, the next largest one, that represents measurements where the FIVC volume suggested full inflation was at the start of test, but the volume that they inhaled was slow. It fell below what we've come to find is a good characterization of maximal effort. Now, being in this wedge, it doesn't mean the data is bad. In fact, most of the data in that segment is probably not bad at all, but we know embedded in there is likely to be some data that represents submaximal inhalation error. And that's problematic because now instead of high confidence, you're back to the 2005 standards days where you have no idea whether or not full inflation was achieved or not. And you can say the same thing for those 2 blue wedges. The dark blue represents where the FIVC volume was less than 90% of the FVC. And that means it's probably not useful or isn't useful for evaluating full inflation. And the light blue one is where the FIVC just wasn't done at all. But again, because there's a lot on the table every time a patient comes in for a visit, sponsors and indeed us at Clario know that it's likely that a lot of that data is good. So we're not throwing that data out. It just remains in there. And so we haven't been able to clean up the problem that Phil's tools point to and what I've shown you in the previous measurements. Now, the red wedge is where the FIVC has done its job. It detected submaximal inhalation error, and in this case, it's about 9% of all the measurements, and that's about what we see consistently across studies. And so what our goal at Clario is to get that, to increase the size of the green wedge. And how do we do that? Next slide, please. So it really comes down to changing the minds of the people that are collecting the measurements. So we have adjusted our training to really focus in on more than just this is how you do spirometry, but instead give really good pointers to those who may have developed and used for many years methods of determining when to give the command to blow that really don't align with best practice. Next slide, advance a little bit. We're going to see this is what they should be looking at when they're, this is what I call the goofy sign. Everybody's got their own goofy sign. And what it means is that when you go to full inflation, the last 5% of that inhalation, things change in the patient's face. And so you really need to be looking at the patient to appreciate it. And that's much more reliable than looking at the volume time tracing, which, by the way, is going to be there while the patient's resting between efforts. You can examine that all you want. And in fact, that time should be spent looking at the FIVC, asking the question, did I time the command to blow properly, and adjusting it on the next effort. So use that FIVC properly. Don't try to make it match by any means. Just try to adjust the start of the inhalation. So those are the kind of things that we've changed at Clario. And right now I'm going to turn this over to Martha, who's going to tell you more about training at Clario. Martha?
Thank you, Kevin. What a great lead in for my presentation. Next slide, please. So I think everyone is aware, getting the proper data and the accuracy and the quality of the data that we need for spirometry is really essential to the success of clinical trials. And our mission with Clario Client Education is to really create tailored site and participant training that really helps facilitate the acquisition of higher quality data. So we want to do this by reducing both the data risks and improve your compliance, while also minimizing the burden that we have on site staff. So again, all of these risks we're aware of, we've talked about. Having a lot of noise in the data makes it very difficult to determine whether or not we have true drug effects, and it can actually slow down things such as enrollment and again, create a lot of waste. But I think what I'd like you to focus on your attention is that bottom line, really getting that good high quality data is based on the relationship, the collaboration between the site staff who are performing the coaching and teaching the participants how to do the maneuvers correctly. It's really pivotal that they have a good relationship and that they are watching the participants' cues, as Kevin said, to make sure that they know that they're doing the maneuver appropriately each and every time. I'd also like to say that by and large, we're looking at experienced both participants and also experienced respiratory personnel who are doing this. And they've got a lot of experience typically in clinical settings, so they've done a lot of spirometry clinically. But one of the first things that we focus on with our training is understanding the difference between the level of quality needed in clinical trial data versus in strictly clinical data. So we spend a lot of time in the why behind what we're doing when we're talking with our site staff learners, so that they really do understand what it is and why this is so important. And we also, understand as well that to do the data the way we need it, or the spirometry the way we need it to get the quality data necessary, is really putting the participants at the level of physical discomfort, actually. So we're really pushing them to their physical limits. So we do want to make sure that everyone is aware of that, and we underscore that those rationale behind this relationship when we do our training. So let's go to the next slide, please. Now, I don't want to belabor this. You can continue to move forward with the different graphics. Obviously, we've looked at a lot of graphics, and one of the things that we also want to make sure that our site staff are very well versed in after going through a maneuver, being able to quickly look and identify the information that the graphics are and the data are telling them so that they can then adjust the coaching method that they're using with the participants so that they can get that maneuver perfected for subsequent times. And I think Kevin said it beautifully. We want them looking at the participant while they're doing the maneuver. They can look at the data during that interim period, and that's when they should be interpreting and helping to communicate what needs to be done and adjusted for subsequent measurements. So with that, let's move forward. So you may be wondering, is an investment in training really something that's going to reap dividends for you when it comes to the quality of the data? And what I would like to do is just share some examples of case study work that we've done at Clario with our actual clinical studies to see what impact training has had. So on the left, one of the measurements of, we think, efficiency and success is the call rate with which site personnel need to call into our customer care center for additional assistance. We looked at two different studies. We'll call them Protocol A and Protocol B. These were very similar in terms of demographics and clinical applications and so on. And in Protocol A, the approach taken by the client was to have an extremely robust training plan. They, in fact, did site training in small groups, hands-on, at a rate 6 times higher than our comparison Protocol B. What we found was a significant difference in the number of calls that come into our call center from site staff looking for additional help. In fact, I think we had only about a 5% rate from the very robustly trained staff versus a 30% rate that we would see with the second protocol that did not have as much training. So that's a pretty significant jump in efficiencies and the ability of staff to handle any issues internally and move very quickly through the process. In addition, another thing that we looked at in terms of the efficiency of our training is how well the site staff were passing proficiency first time through. And again, when we compare a protocol with a robust training plan versus one with a lesser so, we see that there's actually twice as many people passing, so up to 80% of people at the staff level passing proficiency first time through when we have that very engaging training with them versus a 40% when we do not have that opportunity. So again, that gets your site staff up and ready to work their way with patients and collect that data and get your enrollment going very quickly. So again, pretty significant improvement there. And lastly, also, we wanted to take a look, what happens with the data itself, the quality of the data. In this instance, we had a situation where we had a protocol that started with a lesser, shall we say, less aggressive training plan, realized going through via monitoring that a learning intervention would be very helpful. And we jumped from a success rate of about 59% before intervention up into the mid to high 80% afterwards. So this difference, these jumps that we see in performance with a robust training plan are really quite significant. And I think that this speaks to the fact that the training is truly an investment that you make with your site staff and giving them the tools and all of the information that they need so that they can collect that good data first time through. So with that, let's move to the next slide, please. And how do we do this? So just to give you a little bit of background about client education here at Clario, first I want to start with our people. We have a dedicated team of very experienced respiratory trainers and people with lots of practical experience in this area who are our site trainers. And these people are located globally, so we have them in every region so we can deploy efficiently to really any site around the globe. We also create content that is very much tailored to your particular study needs. So if you have pediatric or geriatric, obviously we're going to modify our materials so that they speak to the specific needs of the study itself. We also have variability and flexibility with our content as well, so that we can expand it or streamline it depending on the experience level of our learners in our classes. So we're very flexible in that regard. We also support our proficiency approach in making sure that people are up, the site staff are up and ready and able to perform the work effectively with the participants as quickly as possible. We do help with the monitoring of that. And as I mentioned earlier, if we need to do an intervention, we can do so. We can be at the ready and come in and give a very targeted and tailored learning intervention so that we can get the performance to where you need it to be. Also just wanted to say that our approach is also multimodal. We have everything from online on-demand e-learning to instructor-led that is done both virtually and face-to-face, and that face-to-face small group can be done at investigative meetings or it can be done on site, whatever works best for the client. And we do have language capabilities, so we can translate into native languages, which is of course very important to make sure that the communication, again, that pivotal collaboration between the site staff and the participant is being done in a way that is easy to understand so that we get the performance that we need in order to get that good solid data. So with this, I believe if we move to the next slide, I'm going to hand this back over to Todd to give us some key takeaways.
Thank you, Martha. And thanks all for these presentations. I hope the audience finds them helpful in terms of their goals towards the highest quality data coming from their not just respiratory trials, but trials that may include respiratory endpoints, even as safety. But just to summarize this excellent presentation, let me give a few key takeaways. And frankly, I think we'll just read through them. First, centralizing data collection and application of risk-based monitoring techniques can help optimize the emerging quality of respiratory endpoints without undue burden, especially pointed out by that Tufts report. Data quality in clinical trials is variable, and relying on just the ATS-ERS standards as the only quality criteria leaves studies at risk. So data plausibility assessment can mitigate this risk and should certainly be considered. Submaximal inhalation error is common. It's a major cause of variability and should be the focus of training and surveillance to optimize data quality. Proper performance of that phase 4, the FIVC, is key. As Kevin pointed out, this was new in 2019. It struck me as he defined this as, oh, it's new, as one of the excuses as to why not to do it. I just want to point out, we are currently in 2026. This thing has been available since 2019. So an important thing to consider and certainly to implement in your spirometry measures. And then finally, investing in robust, tailored training focused on practical skill development and study-specific problem solving boost learning retention and knowledge transfer. And with that, I believe we actually do have a few minutes, maybe for some Q&A. And so let me turn it back to Ryan to host that piece. And I guess the other speakers, please come on camera here, please.
Yes, well, thank you all very much for today's very insightful presentation. We do have some time, and we'd like to invite the audience to keep sending their questions or comments for this Q&A portion of the webinar. If you're attending via GoToWebinar, please use the questions window, and for those watching on LinkedIn Live events, feel free to post your questions in the comments section. Now, I've already received some questions from the audience, though, so we'll get ourselves started with those. And the very first question that I have here wants to know, what if the patient is too sick to inhale the FIVC rapidly?
Ooh, I'll take that. So can you hear me? Yeah, okay. So I came from an environment where we had a lung transplant program, and as a result, 30% of our main campus business was pre-transplant evaluations. In other words, the sickest of the sick. And while it's true that some patients are too sick to do good quality spirometry, the vast majority of those patients were able to do it. It was part of the workup. It was spirometry, lung volumes, DLCO, blood gas, 6-minute walk. We really had to put these, it was terrible to do, but it was necessary. And most were able to do a good quality FIVC. However, to say that everybody could do it would not be correct. There are some patients that are just too sick. They don't have enough gas in their tank to finish a good forced exhalation, and then they're so dyspneic that you really are obligated to allow them to come off and resume supplemental oxygen. So, but in the vast, we actually did a poster on this looking at moderate to severe asthma and found that less than 1% of the patients were able to demonstrate that they could rapidly inhale to full inflation.
Yeah, a lot of those you talk about, Kevin, I mean, they shouldn't naturally be in a clinical trial anyway. So, you know, of the normal population we're likely to be enrolling, it's going to be pretty minimal, isn't it?
Did I say less than 1% could do it? It was less than 1% that couldn't do it.
Yeah.
Just gonna jump in and correct that.
Yeah.
Perfect. Thank you so much for that answer. Next up here, I have a question asking, what would be the benefit of running implausibility checks on a typical study?
Oh, yeah, I can take that. I mean, so again, the core problem, if you're just relying on ATS-ERS criteria, you know, you're leaving stuff on the table, as it were. So the only way you're going to find out if there's a problem with the data is really once you've unblinded the study and things start not to make sense. So if you're, you know, these issues are hidden if you just look at the historical way we used to, you know, evaluate trials. So this is the core, you know, benefit. If you find out early, you can actually step in and do something about it. So it may be retraining of a site. In some instances, the data may be so bad, you may want to stop the site recruiting because they're not going to be contributing actively to your study at all. They're just, you know, effectively damaging it. So yeah, so, you know, finding that out helps you to do something about it, either, you know, prospectively for future patients, or you can even do some work on your SAP to try and minimize the impact of those bad patients. So, but we're happy to run through that.
Perfect. Thank you.
And that's, I'll just add, that's critical to the power, right? I mean, the key is to identify this early enough, as Phil points out, that you don't impact the database, right? You need that data. You've defined power based on quality data. And if some measure of the actual data in the database needs to be removed or reconsidered, then you really are impacting power. So the earlier you can identify this and the earlier you can intervene, the better to prevent that kind of impact.
Perfect. Thank you. Now, as somewhat related question, someone here is asking if they can use the implausible data process to detect data quality issues at the time of testing.
Oh, yes, you can. So this is a very interesting area. So, I mean, so eventually what will happen, I mean, we're developing AI tools that actually start to integrate this. I mean, the future vision really is that a lot of those checks can be done at the time of testing and can actually allow you to rectify the testing before the patient goes away. I mean, at the moment, you know, some of those tools can actually also be used. I mean, I talked about biological plausibility of data. So you can actually, just with one data point, you can start to check the integrity of the data and then actually, you know, repeat a test at clinic. So, I mean, it depends on the way the study's set up, but if you can identify that there's an issue with the data, you can then repeat it and replace the problem data. So you're not just looking retrospectively at data, you can prospectively modify the data quality. So there's a few ways in which we can do that as well. So it is very helpful.
Perfect. Thank you very much for that answer, everyone, and for, of course, the presentation today. But we have reached the end of our time. Now, if for those who are interested in hearing more from Clario, you can register for their next webinar, Neuroscience Drug Development, How Novel Applications of AI Could Meaningfully Disrupt Traditional Development Strategies, which is scheduled for Wednesday, May 13th. You can use the QR code on screen and also a link that I've sent in the chat box. And also note that if we couldn't attend to your questions, the team at Clario will follow up with you. Or if you have some further questions, you can direct them to the email address that I'm going to put up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen as you click the exit button, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've also shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching on LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and then gain access to features such as viewing that recording and more. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thanks all.